All right, this is ESM 105. It is September 19th, 2025. Recording, yep, okay, good. Microphone's working. So, we are now talking about natural selection and evolution. Last time we talked about adaptation versus acclimation. I drew the evolution umbrella, and underneath it I put natural selection, artificial selection, sexual selection, genetic drift, four forces that can all lead to changes in gene frequencies, changes in allele frequencies from generation to generation. And if those changes in genetics make the population better suited to its environment, then you're looking at adaptation. Remember though, evolution is any change in allele frequencies, so it does not have to be adaptive. It could be a change in genetics that does not make the population better adapted to its environment. Natural selection, though, will lead to adaptation. All right, this is the only type of evolution which consistently leads to adaptation, because individuals with genotypes better suited to their environment are being selected for. Very good. Okay. We're going to move on and talk about tolerance ranges. This makes sense as a natural transition from acclimation and adaptation, because now we're talking about why species can live where they live. All right. So there are these environmental factors that determine where organisms can live. These can act as selective pressures. Remember that term, okay? A selective pressure is something like high salinity soil or low nitrogen availability, something that's going to allow certain genotypes to do better because they can tolerate that environmental pressure. So it's a factor that selects for certain genotypes. It's a selective pressure. Of course, the selection actually acts on the phenotypes, the physical expression of genotype, but that is determined by genotype, so everything flows through. Okay? Good. Physiological stresses, okay, like moisture, temperature, pH, light, nutrients, competition, predation, parasitism, disease, all right? These are all things that determine where a species lives, okay? Sometimes we've got just pure luck. An organism can't live someplace that it never arrives, all right? And an organism may be present in an area, but then it's eliminated from that area because of bad luck. All right, the tree falling example, or flood, or a random lightning strike leading to a fire that kills a species. Okay. Luck leads to evolution by genetic drift. So remember that phrase of genetic drift. That's when the allele frequencies change, so it's still evolution, but it's not because one of the genotypes was better suited to its environment. It's just some of the alleles got eliminated, and now the allele frequencies have shifted. Cool? Question?
What's the scientific definition of luck?
Oh, yeah, that's a good point. Yeah, very low. Yeah, very low p-values. I don't have a great answer for that, all right? But when we're talking about luck, we're talking about a random chance, like a random event. It didn't happen because you did something. It didn't happen because the organism was a certain way or did some actions that led to this. There was just a luck, involved. Yeah.
So evolution can just happen randomly then?
It can happen because evolution itself just means shifts in gene frequencies in the population, allele frequencies, right? So if you've got a population, I did the beetle example, right? All the brown beetles are here, all the white beetles are over here. A tree falls and it kills all the white beetles. Now the gene pool has shifted so that there are more brown beetle genes. So that population's evolved because it's changed its gene frequencies, it's changed its allele frequencies. All right? Cool. Awesome. Oh, and when that happens, it's called genetic drift. Cool. it's really difficult to break the habit of thinking of evolution as you're evolving toward something or you're evolving to be better suited to your environment. That's not always the case. Okay, tolerance ranges, tolerance limits. There are certain conditions under which species can persist. Sets of environmental conditions, niches, right, if you've heard that concept already. So right here, this is on the x-axis, any environmental gradient. It could be moisture, it could be humidity in the air, soil moisture, maximum temperature in July, all sorts of these different things, okay? But there's, within any environmental gradient, there's a range where the species can occur. It's got an optimal range of conditions under which the species is expected to be most abundant. It has these zones where the species is under physiological stress, but it doesn't kill it, and the species is expected to be less abundant under those conditions. And then, if you exceed the limits of tolerance, the species won't occur because it can't tolerate the environmental conditions. A pretty general concept, right? You've got an ideal average temperature for a plant, and if the temperature is ideal for a plant, it's going to be more abundant than if the temperature is closer to the extremes, moving away from that ideal temperature. Reasonable enough? Okay, very cool. Critical limits, all right? With critical limits, we're talking about the point where the species can't occur anymore, all right? Determining where a species isn't usually as simple as looking at a single environmental factor. Just average temperature won't determine a species distribution, yes?
Is this one of those things where, like, as you spend more time closer to the critical limit, like, the species may adapt so that their limit kind of shifts?
That's 100% correct, right? So species that exist closer to that critical limit, now they are under stronger selective pressure. So if it's temperature, right, and this is a very low temperature, and this butterfly species can't tolerate it. Species that live in the habitat where they're experiencing the low temperature are under a strong selective pressure to develop cold resistance adaptations. And if that happens, then it can become an entirely new species over time. Or it can become a subpopulation, or something called an ecotype, which is a which is basically a subspecies adapted to a specific ecological condition. You can have like a cold adapted subspecies of this butterfly. Right? But they're the same species as long as they can still interbreed. Cool? That's exactly one of the things here. So it's really important, right? If you look at, say, standard issue with climate change, right? You've got plant population, it's spread here. I'm going to draw, there's California. That's the top of California, pretty flawless. And there's a plant population right here, okay? So conditions are changing so that these individuals down here are going to be under more and more stress as average temperatures increase, right? The conditions are going to shift so that the species could tolerate conditions here in 15 years, but can it move fast enough to get there? All right? Species at the edges of the tolerance range, okay, tend to be better suited to moving, if that sort of makes sense, okay? They're also less dense. There's a whole thing about assisted migrations and concepts like this that all stem from our discussion of tolerance ranges. Okay, cool. Good, what do we got here? Sometimes there is a specific environmental variable that's very important in determining a species range. For example, you could have a species that's very sensitive to lead contamination. All right? So this species has a very narrow tolerance range for concentrations of lead. And that's sort of a critical factor in determining that species distribution. Now, when you see a species like this, it might be a good candidate for an indicator species. So some species can serve as good indicators of environmental conditions, like a species that requires a very specific oxygen content in water. Okay, the example right here, trout require cool, clean, well-oxygenated water. So if they're present somewhere and then you survey and they are no longer there, that could indicate that there's pollution, or there's less oxygen, or it's not as clean, or it's warming up. You see, you can use tolerance ranges. You can use species with well-understood tolerance ranges to indicate something about the environment. And that's the idea of bioindicators. And we'll go through a specific example using lichens, all right, that I think you'll all enjoy. Okay, great. So niche theory, right? Niche theory says there are a set of environmental conditions under which species can occur, okay? There's some combination of environmental conditions necessary for a species to be able to exist, all right? Some combination of climate, soil factors, competition, etc., all right? So niches usually are discussed around abiotic environmental conditions, however, just to be clear. So you talk about a fundamental niche versus a realized niche. I'll draw on the board again. Where's my chalk? Where's my chalk at? I had it. Yeah, I'll use this one. Okay. Here's California again. It's pretty good. Flawless California. Okay. You've got a species, and it could theoretically live anywhere within that range. Based on its biological limits, the species has a fundamental niche of existing anywhere within this range, okay? But it actually only occurs here, here, and here, all right? So these patches where the species actually occurs, that is the species realized niche. The species fundamental niche is anywhere where it could occur. All right? So niches are important. It helps us to, if you understand a species fundamental niche really well, you can kind of understand where species are likely to compete and interact. When two species share a niche like perfectly and have overlap, they're likely to be competing for resources, even if the resources are just space. But if they're animals, maybe they're competing for food resources as well as space. So pure or total niche overlap it usually leads to like stronger competition, all right, when the fundamental niches purely overlap. But if there's just some overlap in the fundamental niches, well then actually what I'll say is selective pressures are going to try to push those niches of those species into shifting so there's less overlap, so that the species do not have to compete so much for resources. Question?
So is what you're saying saying that coexistence really doesn't exist?
Oh no, coexistence exists, yeah.
But like, there's always going to be some form of competition.
Competition isn't mutually exclusive to cooperation.
Or coexistence.
I guess that's a good way to put it, yeah.
Absolutely. Yeah, so competition is natural. It's probably going to occur. It's usually not such a strong pressure that both species go extinct, but competition can be a selective pressure. And if there's a high amount of competition, then the species are likely to slightly change, especially the one that's worse at competing is likely to change its niche a little bit and get specialized, get like niche specialization, a concept which we'll talk about in just a moment. Solid, awesome. Yeah, of course. Good. So the ecological niche lets us, by adding that word ecological niche, okay, it lets us focus specifically on the environmental factors. All right, this is the fundamental niche, where it could possibly exist. Are we cool with the fundamental versus the realized niche? The realized niche will always be a smaller amount of space than the fundamental niche due to biotic interaction. There's always some sort of competition or human activity, development, things like that. Okay. Niches are often visualized like this. as little diagrams, as little circles within a diagram like this. So this species can exist anywhere within this temperature range from here to here, theoretically. It could also exist anywhere within this moisture range from here to here. But when temperature and moisture are very low, this species cannot exist. So what you really end up seeing is they call it an nth dimensional hypervolume. It's really weird as a term, okay? But you add more and more variables to determining the niche. So it'll be temperature, and then moisture, and then light availability, and then there's disease pressure, and all these other things. So once you add like 7 or 8 different variables into determining the niche, then it has n dimensions. It's no longer 2-dimensional or 3-dimensional. It could be like a 9-dimensional shape, which we can't visualize, okay? But the idea here is that there are a lot of factors that interact to actually determine the fundamental niche. And usually the convention for teaching this is the fundamental niche is determined by abiotic factors in the environment, and the realized niche is determined by biotic interactions. So the species, if it was the only species, would live throughout its fundamental niche, but because there are things like competition, I'll stick with competition for now because we're going to talk about it more, because there are things like competition that limits these species to only occurring within a subset of those conditions. All right, fundamental versus realized niche. And then here, as niches overlap, competition is likely. And also, if there is a habitat that is vacant because nothing has a fundamental niche that fits that space, it is likely that that space will eventually be colonized by a species that will adapt to fitting into that niche. That's that concept of nature abhors a vacuum. If there's a pool of resources somewhere in the environment, something will adapt to access that pool of resources, even if it's an incredibly challenging habitat type and nothing currently shares a fundamental niche with the conditions there. How's this working for people? Are we feeling good? Okay, awesome. So yeah, if niches overlap, more competition. There's this model called what do they call it? The snowballs on the barn model. They call it a model, not a martle. Okay? So now we can visualize, theater of the mind. Welcome. Oh, it's so cold. Brr. Look at that barn wall. I know, we're all feeling it. I scoop up a snowball and I throw it at the barn wall. And that snowball sticks to the barn wall. It would make a lot of sense for me to do this on the board where I could draw the snowball instead of purely having you visualize it. All right. Okay, cool. Yeah, it was fun. Yeah, let's just do that then. You know what? You've convinced me. All right, throw the snowball. It hits the barn. It sticks. All right. I throw another snowball. It hits here. It sticks. All right. The barn wall is the set of all environmental conditions. Okay. And when one of these hits, that's the niche of a species. Now, if another snowball hits on that spot where that snowball already is, it can't stick there. All right. It falls off, because that niche is already occupied. So niche overlap, if it's too intense, competition will cause niches to shift. And that's the snowball on the barn wall model for explaining niches. Was that? Oh yeah, snowball. I'm gonna try. Snowball on the barn wall. All right, cool. Yeah.
So wait, but can't overpopulation happen? Like where but I guess that expands the niche instead of like overlaps it.
It's more, so you can have overpopulation. So I'm gonna sort of make sure I'm with you. So you've got a population that exists in its realized niche. And then if that population becomes overpopulated, that can be an internal pressure, right? The population should reach a carrying capacity and stabilize and sort of go like up and down in population size there, right? That's the idea. That's the model. What the model tells us there. It can also expand if it becomes overpopulated, and spatially move outside of that niche. Or if there are too many individuals, now there's a pressure to move out from that niche into the margins of your fundamental niche, where you don't have full tolerance for all the conditions, And now there's a pressure, selective pressure, for the individuals that migrate out to adapt to those conditions. And the population can expand spatially. So it's easier for populations that have a broad range of tolerance limits to expand their physical range, their physical amount of space they take up, right, than species with a narrow one.
So it's like throwing a large snowball.
Kind of, but the trick here is that this isn't space. This isn't like a portion of California. This is a set of environmental conditions, which is really tricky because when we're talking about niches, it's like, oh, the species has a niche in this area. If the species expands its tolerances, it could expand the niche that it could occupy and maybe expand its physical, like spatial range. But they're not identical they're not the words don't mean the same thing. So even though they're super tightly connected. Satisfying enough?
Yeah.
I love it. Awesome. Okay, cool. I want to get into ecological indicators. So we had to talk about tolerance ranges and the niche theory and niche concept in order to talk about ecological indicators, okay? And then we will talk about a concept that competes with the niche theory, even though it's usually taught as just an entirely separate concept. It actually is, both of these are developed, the niche theory and the other thing, no spoilers, all right, are developed to help us predict where species will occur, okay? Predict which species will be dominant. So that'll come up in a moment. But we needed to talk about niche theory to get into these ecological indicators. And really we needed to talk about tolerance ranges. Okay. We're going to go with bioindicators, ecological indicators that are alive. We already talked about the trout. It needs a certain set of conditions in the water. If it disappears from that habitat, then the conditions might have shifted. We'll use our lichens. It's a nice example. So biosurveys. In a biosurvey, you survey animal, plant, microbiological, some sort of community, or you survey for a specific species. There needs to be a very standardized method, all right? So not every survey that you conduct is just to keep track of what the community looks like in a given area. Some surveys are used to determine if there's an environmental threat, if there's like contamination. So if you're doing a project that, you're doing a power plant project, and there's a chance based on previous work that this could contaminate streams if they're not treating wastewater correctly. So in order to keep track of that, regular surveys are done for amphibian abundance in that stream. If the amphibian abundance goes down or the amphibian diversity goes down, that's an indicator, an ecological indicator that there's been contamination in the stream, and that triggers the next steps in testing and surveying. So biological indicators, using ecological indicators, is advantageous because it can be faster sometimes, or simpler, less technical. Good. What's a biotic index? Okay, this is pretty cool. So that you don't have to, but in order to conduct biosurveys like correctly, in a codified way where you can deliver a number, and if that number falls below a certain value, that triggers a response, which is how these regulatory systems like to work. They're like, you do the work and deliver a number, here's the protocol. If that number is below 3, then we'll go ahead and implement corrective actions, okay? It's not a very holistic outlook. So they use these things called biotic indices. So you'll get a biotic index. Where's my example? I have a good one here for the lichens. It's a little clunky. This could be a little less clunky. Whatever, we'll get to the biotic indices in a second. I'll tell you verbally, and then I'll show you an example, okay? So you go out, you do a survey, you get the abundance of all these different species. If the species is present versus absent, you mark a number down, all right? Each of those numbers is weighted, where the presence of one thing means more than the presence of another. and you push all those things through a little model, all right, through a little equation, and that spits out a value. Depending on the biotic index, it could be like from 0 to 1, or it could be from 1 to 5, or something like that. All right? But that number indicates something about the environment. So there's like a lichen biotic index for looking at air contamination. You survey specific species of lichens, you say how abundant they are, all right? And then you use that to calculate a value that then indicates whether or not there's air pollution. Question? Yeah, it's like, well, the cool thing about this is if the habitat is of interest, there are probably regular surveys going on to just catalog the species there, right? Or if the habitat is likely to be under pressure, or if it's a protected habitat. So you can take the data that you collect from the surveys and just shove it into the calculation for the biotic index. But so it's like, yeah, it's like a step 1 and then like a step 2A, step 2B, step 2-3, 2C. You have all these different ways you can use the data. And one way is to calculate biotic indices. All right? So sometimes you have single species methods where it's just like salmon's present, okay, high oxygenation, done. Stinging nettles present, high soil fertility, boom. One species indicates a certain environmental condition. A biotic index encapsulates or keeps track of multiple species to get a more nuanced picture. Does this make sense so far? The index incorporates multiple species. It's a multi-species approach. So here we go. What do we got here? What can we use indicators for? All right, this table, I put it in here, and I was like, this table's good. Now I'm looking at it again, and I'm like, it's kind of a... What this table tells you is the sort of things that biotic indicators can be used to assess, right? You can assess pollution levels, oxygen levels, soil properties. So like oxygen levels, if you see species that can tolerate low levels of oxygen, and you couple that with the absence of species that require high oxygen levels, that would indicate that the oxygen levels are probably low, because the conditions have changed so that only the species that tolerate the low oxygen levels are present. Does that make sense? Cool. So it's usually a combination. Those biotic indices are stronger. Sustainability, species richness, there are biodiversity patterns that you can indicate. And this is basically just like how many species are present and what's their relative abundance. Yeah?
So if you have 3 species in the environment, they're all environmental indicators of oxygenation,
Mm-hmm.
per se, in a water body, and 2 of 3 of them are less abundant than expected, then you get like a score. Is the biotic index the score, the 2 out of 3?
Essentially, yeah.
So 1 out of 3 oxygen.
Yeah. Sometimes it's as simple as you've got this sheet and you've got the low oxygen, medium oxygen, and high oxygen species. And if they're there, you put a 1, and if they're not there, you put a 0. You average the values, and then you average across all of them, you know? And that can give you the, yeah. Okay, let's use a lichen example. This is, I think, pretty fun. Single species versus multi-species approaches. Usnea cornuta, all right? You see it all over the place here. It has a sort of a narrower tolerance range where it requires clean air. It's a pretty vague statement, okay? It also cleans the air. It's beneficial in that way too. So it's really only abundant in areas that have what we would just classify as clean air, okay? Just because it's absent does not mean the air is polluted, but if it is present, it means the air is clean. So you see, you have to know what the species actually indicates, and its absence, just the absence of a single species, doesn't necessarily tell you that the environment doesn't meet the conditions it needs. It could just not have arrived there, or there could have been some random event that removed it all. You know, maybe some model train enthusiasts came through and harvested all of it to use for shrubs in their model train sets. You know. So here's a more complex approach involving, it's going to involve multiple species, okay? Here's another species, Hypogymnia phasotes, all right? It can take high levels of sulfur. Now, there was one species that we're looking at here, and we're saying, oh, it's present. So if it's present, that doesn't necessarily mean there are high levels of sulfur. It can just tolerate high levels of sulfur. It could be surviving in an area that doesn't have high levels of sulfur. But if it is present and other species of lichen that can't tolerate high levels of sulfur are absent, then that indicates there may be sulfur contamination. So it does require that you know how abundant is this species relative to all the other species. But getting a little more nuanced here, it doesn't actually mean you need to be able to identify all of the other species. If this is the only species you can identify, you just do a survey for lichens, and you say, and you have to ID this one, and then all the other lichen species are species A, species B, species C, species D. Because what you're trying to calculate and represent is that this species is at a high abundance relative to all the other species of lichen. Okay? So you see how that's more complex. It's still single species because you don't have to ID the others, but you have to know about, it's a multi-species, single species sort of overlap here. All right? All you're going to be calculating and presenting is the relative abundance of this species, saying that it is very abundant relative to the total lichen population. All right, and I'm going to explain the term relative abundance because it's incredibly important in ecology. Cool? So this is not a full like multi-species approach where you have multiple bioindicators that you're using all at once. You're using a single bioindicator, but instead of just looking at its presence versus absence, you're looking at its relative abundance. How's that working for people? Questions? Awesome. So, relative abundance. First, let's get rid of the typo. Okay? So, we will talk more about this in our next PowerPoint, which is all about populations and communities. Describing diversity. All right? When I say the word diversity, let's go with plant communities, because I am a plant biologist. All right. If I describe a community with high diversity, all right, what does that sort of make you think? We got one, yeah.
Multiple species, an abundance of multiple.
Awesome. Many species. Does anybody else have something that high diversity makes them think of? Yes?
Thriving ecosystem? I
A thriving ecosystem? Okay, cool. Yeah, great. Anybody else have something about a high diversity community they want to throw out there? Yes. Just give you a turn and we'll pop it over to you. Yeah, it'll come back to you, I promise. Yeah, yeah.
I was gonna say maybe less disturbed.
Okay, less disturbed? Yeah. I love it. You got one still?
Diverse abiotic factors.
Cool. Diverse abiotic factors. Maybe even like heterogeneity in the environment. Yes, I saw another hand. Where was it? Yeah. Ah, you stole. Dang. All right. So we got high diversity. These things are accurate, right? One measure of diversity of a community is the number of species. A lot of species indicates a high species richness. So this is sort of the most This is one of the most common, it's the easiest to measure in doing a survey of a community for diversity. That's species richness, which is usually represented as a capital S. All right? So we've got some other things. These are generalizations more about the benefits of a high diversity community. A high diversity community is a thriving ecosystem. It's usually, if there's a lot of, it's a less disturbed ecosystem, and there are diverse abiotic factors. Now, these things are not always necessarily true, okay? Just because a community has high diversity, it could be overcrowded with species, right? There could be too many species with too much niche overlap, and none of them are actually at a very high abundance, and the community could have very low resilience, and it could be prone to collapse, all right? But we usually can generalize and say a higher diversity community leads to a more thriving ecosystem. That's the generalization. Less disturbed. Well, if you exclude disturbance from an environment entirely, like through fire suppression, right, then you can actually get a much lower diversity of species. So this one is a common, it works sometimes, and then it's also a common misconception. Like if we want a high diversity community, disturbance usually encourages, if it's not too extreme and it's regular and minor and it's natural, it usually encourages diversity because it opens up niches, it opens up resources, right? Diverse abiotic factors, absolutely, right? If there's the high diversity in the soil conditions, for example, that allows a lot of different species to coexist in the environment, in the community. So an environment that has a lot of heterogeneity or isn't totally, consistent across everything, is likely to harbor high diversity, okay? Now, we've got one measurement here of diversity, though. And if you're going out and actually trying to survey for diversity, well, you need more metrics than that, besides just the number of species. Because the number of species richness, doesn't necessarily tell us everything about a community. Let's look at two communities here. All right, these are two different communities of plants. They both have the same species richness. Both of them have two different species in them. But they have different relative abundances, and they do not have equal diversity. So let's go out on a limb here. Which community would you say is more diverse between these two? Community A or community B? It depends on your diversity measurement. So if species richness is our measurement, then they have the same diversity. But if we use any other metric of diversity, then species B becomes a more diverse community because there's not one species dominating, all right, the whole community. This requires an understanding of relative abundance. So the relative abundance values here in the different communities, we've got 0.1 for the purple, 0.9 for the white. 0.5 for the purple, 0.5 for the white over here. So the relative abundance value just tells you what proportion of the community is made up of each species. And whenever you're using one of the models for giving you a diversity value, like the Simpsons index or the Shannon index, or one of the other indices that I forget every time, because those are the two that we mainly use, all right? You are using relative abundances, okay? Do we see the difference between these two? Awesome. So sometimes you just want a number that represents how diverse a community is. Usually you get that by taking the relative abundance values and mathematically transforming them and getting some diversity index value. And if it's higher, the community's more diverse, and if it's lower, the community's less diverse. It makes life easier for somebody interpreting the data. Yeah?
And with finding that diversity, can you like further like maybe diagnose the health of the ecosystem or?
Yeah, absolutely. So when we're using when we are using more nuanced metrics of diversity, like things that incorporate the relative abundance, we can make more accurate generalizations. Like it would kind of make sense that this community might be more stable than this community, because if something stochastic happens and that gets destroyed, now the diversity just plummeted,
Yeah.
right?
When I was thinking of that, when I was thinking of this comparison, I was thinking like, oh, the one on the left is like maybe like an invasive species like taking over or something.
Yeah, absolutely. So that's great. So using a more nuanced measure of diversity lets us make generalizations that are better, but more well-founded, right? Yeah. So species richness, relative abundance is super important, and usually, like when we're doing, let me just show you real quick. Actual measurements of species diversity. Let's just go with a Shannon index calculation. They look really complicated. But honestly, they're not that bad. Check it out. This just has relative abundance of each one of these species. Each one of them is 25% of the community. And so to calculate the Shannon index, which is one of the most commonly used ones, at least in plant biology, because it does better with percent cover, concepts that we'll talk about later. Okay? The equation is just take the relative proportion for each species, multiply it by the natural log of the relative proportion for that species, all right? And then do that for each species, and then add them all together, and then multiply it by -1, and that gives you a number. And now this is the Shannon index. You can do the whole thing in Excel in 90 seconds if you remember the keyboard shortcuts. So you need to understand relative abundance, but you don't have to memorize how to calculate Shannon indexes or anything like that. So how do we feel about these two? This is, I feel like for ESM 105, this is good. We just talk about species richness and relative abundance because this will lead to all the other measurements of species diversity. Okay, cool. So next week, we'll come back and we'll talk about this really cool air quality measurement, this multi-species approach protocol for lichens and measuring air quality. We're doing great. Competitive exclusion, resource partitioning, how speciation happens, Jansen-Connell, mimicry. We got a few slides to get through, but don't worry. We just need to get through it by Wednesday, the end of Wednesday, and I'm confident we can do it. Keep up the good work, everybody. Hey, don't forget, it's Friday, so that means that there's a discussion. I'm really appreciating everybody's work in these discussions. Keep participating, please. Respond to somebody else's, put your own information in there. I respond to some of them, It doesn't mean that yours was bad if I don't respond to it, so you know, but I'm responding to some. Have a great weekend, everybody. We're done. Enjoy the rest of your day.